Now, we've talked about the naming and the structure of these simple peptides. And one of the things we reviewed in the last lecture was the isoelectric point and pKa's surrounding our amino acids. Now, we talked about how amino acids do have multiple pKa's, and some of them have additional pKa's outside of the ones that are attached to the amino group and the carboxyl group. And those are based on their R groups that they have. We also talked about kind of what these would look like in a buffer and when they are charged and not charged based on the pH that we are at. Now, how do we figure something out like this, but for a peptide? We talked about finding the isoelectric point of an amino acid with multiple pKa's, but what about a peptide? A peptide has a ton of pKa's that are there, and we will approach finding the pi, the isoelectric point of a peptide, the same way that we do with an amino acid. And again, the key is you have to find the point where the charge of the molecule is 0. The pKa before and after that point, you will take the average of, and that is your pi. So for something like a peptide like this, you need to use a table with pKa's because we need to know all of the pKa's of each of the groups. Here's the table that I presented in our last lecture. This is the table that has all the pKa's that you will need for amino acids. It tells you, again, the pKa of an amino acid for the first one is for its carboxyl group. The second one is for its amino group. And the last one is for an R group if it is ionizable. Some of them, many of them, do not have R groups that ionize, but for the 7 amino acids that do have groups that ionize, they've been listed. So first, we want to think about, for this particular structure, which of these pKa's are important. Well, we already know the peptide that I brought up is this GDE. It's a glycine and aspartic acid and a glutamine, or a glutamate, excuse me. This one is going to be related to the pKa's that have been highlighted. So G in this case is glycine, and you can see I've highlighted in red all of the pKa's that are related to glycine. D, our aspartic acid or aspartate, is highlighted as well in red at the bottom, and E, glutamic acid or glutamate, is highlighted in red at the bottom. Now we have to think about which of these pKa's is pertinent to our peptide structure. What do I mean by that? Well, again, I have the structure here, and I've highlighted the pKa's that are pertinent, and I've just limited it down to the amino acids that are important for this structure. So in this structure, again, this is GDE in that order. We start with a glycine. The glycine has the amino terminal end on it. So the glycine amino pKa is important for this structure. Notice on the other end, E, this is our glutamic acid. The pKa that is important here is the carboxyl end. So the glutamic acid has the carboxyl end of the peptide structure. Within this structure, there are also two amino acids that have R groups. And so those two R groups are also important. So again, you want to find the amino acid that has the amino end. So the pKa of the amino acid with the amino end should be highlighted. And the pKa of the amino acid with the carboxyl end should be highlighted. And lastly, any pKa's for R groups of those amino acids should also be highlighted. So, based on what we have here, we can see some information that is pertinent for this particular peptide. Here I have the structure of the peptide shown again, and I have limited our table to the 3 amino acids that make up this structure. Now I've highlighted the amino acids to represent each pKa that is important to our structure. So you can see starting at the end of our structure with glycine, glycine is the amino acid that has the amino terminal end. So the amino group of the glycine is important for this peptide. This amino group will have a charge on it. And so for glycine, I have highlighted the pKa associated with its amino group. Now, for our end terminal, our carboxyl end terminal of this particular peptide, we are going to be using glutamate. So the glutamate part of the peptide has the carboxyl end. And so the pKa associated with the carboxyl end of the glutamate amino acid is important for this peptide. The other two highlighted pKa's are pKa's for the R groups, and both of these R groups are in our amino acid. We have the aspartate and glutamate amino acid, and their R groups are present. So the next step to thinking about the PI of this peptide is to order the pKa's based on increasing pH. So you can see the first pKa is this pKa associated with the carboxylic acid. It has the lowest pH at 2.19. The next one is this pKa for the aspartate R group, then the glutamate R group, and then last, the glycine amino group. So I've labeled these in increasing pH, and I'm going to show you again where these are on the structure. The first one is the carboxylic acid end of our amino acid peptide. The second one is the R group of our aspartate or aspartic acid. That's this group. The third one is the R group of our glutamate or glutamic acid. That's this one. And then the last one is the amino group of our glycine, the amino terminal. So each of these will change with an increase in pH. And I want to think about how the charge of the whole molecule will change, going from the lowest pH to the highest pH. Starting at the lowest pH possible, everything is protonated. Remember, as we are going to increase the pH, as we get more basic, I'm going to one by one remove a proton. Each of these pKa's will tell me when that proton will be removed. So all of the groups with O minuses, they're going to start off with their hydrogen. They will not have a minus. And our NH3 should be NH3 plus. It has its extra hydrogen as well. So at this state, at the very bare minimal state, the very beginning state, before we've progressed through any of our pKa's, we have a overall net charge of +1. Now notice this is before any of our hydrogens come off, because each of these groups with the oxygens, I wrote my H here in the wrong place, should be here. Each of these groups with our oxygens are all protonated, and so they do not have a negative charge yet. Now, as we move to the first pKa, passing the first pKa at 2.19, so at a pKa pH of 2.19, the hydrogen on the glutamate will come off. So for this molecule, that is the hydrogen on the terminal of the glutamate. I numbered it already, number 1. This hydrogen will come off and we will have a negative charge. At this point, our charge, our net charge of the molecule is 0. Okay, so we have already reached the point that is our isoelectric point, where it is 0. The pKa after the point at which it is 0. So we have to think about which, we just passed our first one, our pKa of 2.19, in which we have a neutral state of 0. And the pKa that is after that is the pKa of 3.65, in which we will remove the proton from this group on our aspartate, the R group on our aspartate, leaving us with a negative charge here, and an overall -1 charge. Okay, so if we were to continue just to see what would happen on this molecule, and we went to our next, past our next pKa, past our pKa of 4.25, we passed that pKa, this would mean that we would remove this proton here on group number 3, the R group of our glutamate, again leaving us with a negative charge. We'd be at -2. And finally, if we were to continue further and remove the final proton on our glycine on the amino group here, we would lose the positive charge and ultimately have a -3 as our total charge. So again, let's think about each of these transitions. To go from this first state to this state, we had to first pass our first pKa, and this is of that group 1, which I have highlighted as 1. And to go from this point to this point, we had to dissociate our next proton at pKa 2. These are the 2 pKa's that border when our molecule is at a 0 state. The pKa for the first one, our lowest one, is 2.19. And the pKa for the second one is 3.65. So the pi for this molecule will be the average of those 2 pKa's that border when the molecule, the whole peptide, has a 0 charge. And that is how you solve for the pi, the isoelectric point, the pH of the 0 state for a peptide.